Thanks, Justin. And I'd just like to thank Andrew and Rose for organising for me to be in Shepparton this week. I really appreciate it. So I'm here to give a brief update on the research and convince all the packhouse operators at the summit to complete a shorter self-assessment survey that's designed to capture performance of microbial food safety management in packhouses. For those of you who don't already know about this research project, the main goals are to develop a quantitative microbial risk assessment model for the apple industry that will identify the most important risk factors for the major foodborne pathogens, as listed, and determine the effects that different practices have on risk level, which will assist packhouses to apply their resources effectively. We also aim to facilitate risk management through microbial risk communication and the development of risk assessment tools for packhouses. The risk model is progressing, but as with any model, more data result in improved accuracy. I need as much information as possible on the range of industry controls and their parameters and how well the controls are managing microbial risk. An observational study already completed, which included a hygiene audit against best practice, provided the information to set up the model framework, as shown here. As you see, the model encompasses risk assessment through the whole supply chain. The model is being developed, and I apologise, I don't have a pointer to point to things as I go. The model's being developed in a program called Analytica, and this is an influence diagram of the framework. Each rectangle is a module, and the arrows depict a module that influences the outcome of the module it's pointing to. Working on the model highlighted that the evidence for effective food safety management is partly anecdotal and often limited, and it would be useful for the industry and development of the model to have a more objective measurement of control so that control can be easily demonstrated. So we developed an Apple Packhouse diagnostic indicator tool that consists of a questionnaire, and all the answers are listed along the top there, and it was modified from a tool developed in the Netherlands. 6 packhouses have formally completed that questionnaire. Taking the form of an internal audit, it's a snapshot of strengths, weaknesses, and improvement opportunities. And this shot shows how the answers along the top convert into spiderweb or radar diagrams. Comparing just one of the spiderweb diagrams between 2 packhouses for the design of food safety control activities, we can see how a higher score for packhouse 1 on the right is shown by more green infill. And I apologise, I don't know why I put packhouse 1 on the right and not on the left, but anyway. So that more green infill indicates that there's better control in packhouse 1 than in packhouse 3. So there's a really good visual on which specific controls need to be improved. Our experience with completion of the full questionnaire was that packhouses identified gaps in their control and they could see ways of how to improve it. For the 6 packhouses that completed the full questionnaire, overall results compared favourably with the observational study. For packhouse 5, the difference in performance between the observational study and the diagnostic indicator tool was explained by some answers overlapping 2 situations, because those answers include multiple qualitative descriptions. Also, the diagnostic indicator tool is a more comprehensive assessment, so it's more sensitive to system performance. Whilst it's a tool based on technical and managerial measurement, there are questions that overlap with measurements of people's risk awareness, which is part of food safety culture or the food safety climate. Because the questionnaire is about 70 questions, it's too long for most people to have the time to complete, so we developed a 10-minute mini version so that more people can complete it and I can capture a broader range of data for the risk model. So when you do the survey, you'll also be able to identify gaps in control and take action where needed. So this is the survey link, and if you want to go onto your phones, tablets, and start doing the survey now and not listen to anything else I have to say, then please go ahead. It is completely anonymous. The Qualtrics system doesn't capture who's answered the questions. So I'm now going to run through some of the questions of the survey and show you how the data will be used in the risk model. So this is what you'll see when you first go online, and the very first, so a bit of explanation about the survey, and the first question is simply about consenting to do the survey, something the universities insist you have to have. So most of the questions can be answered quite quickly, but there are some that need to be read carefully because there are some subtle differences between the choices. The three introduction questions on certification, customers and staff numbers provide the context of the organisation and the supply chain characteristics. Part 1 questions about netting, source of irrigation water and any exclusion period between overhead watering and harvest provide context to the microbial quality of apples entering the packhouse. And I know that most apples are not watered by overhead watering, but in some orchards there is overhead watering for cooling, which can possibly happen not too long before harvest. So you can see the questionnaire now refers to situations, and each situation is scored with 1 representing the highest risk and 4 the lowest risk. There are now 14 questions about food safety control activities. I won't show all of them, but they include questions about workflow controls, the food safety system in place, and equipment design. Note the options to answer recognise that sometimes parts of a system are well controlled and other parts are not so well controlled. So you need to just choose the answer that's closest to your situation. Sure? Right. It then moves to the ability to exclude animals from the packhouse, the sanitation programs, and the extent of personal hygiene requirements. Once there are 4 situations, many packhouses will be described by either situation 3 or 4, which is improved control, but there are important differences between those 2 situations, so you need to be careful about that. There's a series of questions about wash water, its source, the use and validation of sanitiser, which is an important part of sanitiser use, the type of sanitiser used and its target level, and the frequency of monitoring and adjusting the sanitiser. So that's just the first couple. On equipment, we ask about cleaning of critical potential contamination sites like brushes and conveyor belts, and there's 1 question on risk analysis methodology. Quality assurance activities are covered by 2 questions about microbial testing, E. coli in wash water, and environmental swab samples for listeria. And my experience so far in the industry is that environmental swabs for listeria testing is a fairly low incidence. The final 2 questions ask about use of feedback and the evidence you have of food safety control. I now want to show you just a couple of slides to demonstrate how results from the survey will be used in the risk model. So going back to the model framework, that I had up earlier, the next slide is simply going to show the contents of the food handler module over on the left there. So with the food handler module open, you can see all the factors that influence food handler cross-contamination to fruit and equipment. The green rectangles on the left are what we call decision nodes, and when the model is finished, they'll be active nodes for the user in the packhouse to answer. Survey questions 8, 13 and 23 provide the background and information for some of these nodes on personal hygiene controls. Now this is now the same shot of the food handler module open with the hands to fruit node open. And it's showing that the transfer of bacteria from food handlers' hands to the surface of fruit can be defined mathematically as a normal distribution down there in the left-hand bottom corner. That simply means that some people will wash their hands well, some people will wash their hands poorly, and some people won't wash their hands at all. And we know from experiments done with Listeria monocytogenes how that influences the variation in contamination reduction on hands. So that just gives you a bit of a flavour of what the risk model will look like and how it's going to be used. So I'd just like to emphasise, to finish off, that now I'm focusing on the industry practices that you're going to tell me about in the survey to make the risk assessment model as user-friendly and as realistic as possible. So there's the link again, and hopefully I've provided you with enough incentive to go ahead and complete the survey for me. Now I'm going to be around for a while today, obviously, and I'll be with the group tomorrow. We have a couple of computers up the back, one computer up the back, I think, 2 computers up the back where the link is on those computers, and so anyone who would like to complete the survey can go and see Andrew straight after I talk in the break and later on, and we'll get as many responses as possible. Thanks very much for listening.